Welcome! In this tutorial, we'll explore the relationship between protein sequence and structure using the green fluorescent protein, or GFP, a remarkable molecule that glows under ultraviolet or blue light. GFP is widely used in molecular biology as a fluorescent tag, and here we will use the RCSB PDB website and MolStar 2. Search for and explore a GFP protein structure. Visualize GFP's 3D structure and understand its function. And investigate the GFP gene and its important mutations. To begin, open your web browser and go to rcsb.org. In the search bar at the top, type green fluorescent protein, then select it from the UniProt molecule name suggestions that appear. Scroll through the results and find the PDB ID 1EMA, then click on the structure to open its structure summary page. This entry corresponds to the original GFP from Aquaria Victoria. To begin 3D exploration, click on the Structure button in the Explore, in 3D section below the image, or the Structure tab at the top of the page. This opens the Mol Viewer. At the top of the viewer, you'll see the protein's amino acid sequence. Clicking on a residue in the sequence highlights its position in the 3D model, and vice versa. For example, if you click on CRO65 in the sequence, you'll see the corresponding residue in the 3D structure highlighted, allowing you to explore its position relative to other residues. The default view is a cartoon representation. Helices are curly ribbons, beta sheets are arrows, and loops are lines. Let's now switch to a view that shows all atoms and bonds in a ball and stick representation. Hide existing non-standard and water representations by clicking on the respective eye icons. With the polymer component remaining visible, click on the three dots next to it to reveal a drop-down menu. Then select Cartoon Representation to expand another menu. In the Type section, click on Cartoon to see that the default cartoon representation is selected. Next, select Ball and Stick from the drop-down to switch to a ball and stick view. This will display the atoms and bonds in a more detailed atomic model representation. Again, in the polymer component, click on Set Coloring to reveal another drop-down menu. Select Atom Property followed by Element Symbol. This will color the atoms based on their element. Now, let's get a better look at the protein structure with all atoms and bonds shown. To do this, we are going to zoom into the protein. To rotate the structure, press the left mouse and move, or use the Shift button plus left mouse button and drag, allowing you to examine it from different angles. Zoom in and out by using the mouse scroll wheel. On a touchpad, use a two-finger drag. Pan the structure by pressing the right mouse button and move, or use the Control button plus the left mouse button and move. This detailed view helps you observe how atoms are connected, but it can be visually dense. Let's now isolate a specific region, the chromophore. Start by refreshing the page. Next, hide non-standard and water representations again. Click the arrow icon to enter selection mode and set the selection level to residue. Then, click on CRO65 in the amino acid sequence panel to select the chromophore. This residue should now be highlighted, showing that it's been chosen. Click on the box icon at the top to create a component of selection. This will generate a new component based on the selected residue. Next, click on the Create Later panel to the right of Representation and select Ball and Stick. Click on Options for another drop-down menu. Under Label, click on the right side and name the component Chromophore. Finally, click on Plus Create Component. Click on the eye icon next to the newly created chromophore component in the Control panel to make the component disappear or appear. Rotate the structure to see how the chromophore fits within the overall shape of the protein. This will help you visualize its placement in the context of the entire structure. Now let's take a look at the hydrogen bonds that help stabilize protein structures like GFP. Start by refreshing the page. Again, hide non-standard and water components. Click on the three dots next to the polymer component and then select Cartoon Representation. In the Type section, click on Cartoon. Then, select Line from the drop-down to represent the protein as lines, making it easier to visualize hydrogen bonds. To customize the appearance further, click on the three dots next to Line and select Advanced Options to reveal additional customization settings for the line representation. Adjust the size factor to 4 to make the lines representing the covalent bonds in the structure more visible. To view the additional hydrogen bonding interactions, click on Add Representation in the Polymer Component dropdown. From the drop-down menu, click on Non-Covalent Interactions to display the interactions, including hydrogen bonds, between different parts of the structure. These interactions are shown as dashed lines. To adjust the atom coloring, click on Set Coloring in the Polymer Component dropdown. Next, click on Atom Property and select Element Symbol. Now, we'll add outlines for clarity. Click the Settings control icon, then toggle Outlines to On. This adds a dark contour around each atom. Now, you can rotate and inspect how hydrogen bonding stabilizes beta sheets and contributes to protein folding. Thanks for following along. You've now learned how to use RCSB.org and Molstar to explore the protein sequence and structure of GFP. These skills can be applied to many proteins in the PDB. Keep exploring and happy modeling.